Hi, welcome back to biology. In this video lab, we will be investigating the process of fermentation. Fermentation is a process that prokaryotes and eukaryotes both do, and it's what happens when those organisms try to metabolize carbohydrates, but they don't have enough oxygen available to completely and thoroughly break down, oxidize the carbohydrate to extract the maximum amount of ATP, energy, out of it. So instead, they take a shortcut, which produces less ATP. There are two types of fermentation. Organisms such as us, humans, and some prokaryotes as well, undergo lactic acid fermentation, where the glucose molecule is broken into pyruvic acid, and then that is converted by NADH into lactic acid. What we will be investigating today is alcohol fermentation. So, what happens in alcohol fermentation is the glucose molecule is first broken down into two pyruvic acid molecules. Those pyruvic acid molecules are then converted into a series of other compounds with the assistance of NADH. First, the pyruvic acid is broken down into acetaldehyde. Then the acetaldehyde is broken down into ethyl alcohol, or just simply ethanol, and carbon dioxide. These are the byproducts, waste products of alcohol fermentation. In this lab, we will be making these two compounds, and we're going to use balloons to try to capture the carbon dioxide gas and compare a wide range of different food sources to see if the rate of fermentation is affected by them. Here's what we have. I have 9 beakers. They will all contain 200 milliliters of water. This is our control beaker, which will have only water in it, and then the yeast is inside these balloons. It's October, which is Breast Cancer Awareness Month, so I picked pink balloons. All of these balloons have 20 grams of fast-acting yeast in them, so I just bought these. I bought everything here at Walmart, as a matter of fact. So 20 grams of yeast and 200 milliliters of water will be in each of these flasks, except for the final 2. These 2 flasks contain regular table sugar, 10 grams in this one, 20 grams in this one. These 2 flasks contain flour, which is a more complex carbohydrate, again 10 and 20 grams. These 2 flasks contain artificial sweetener, no calorie. So what's going to happen with that? I want you to try to make a prediction. This flask is going to have syrup in it. Now, I've never done this before. I've done this lab several times, but I've never used syrup. Syrup is really just liquid sugar, if you think about it. And I looked at the label and I did some very simple math. This beaker will have the equivalent of, let me double check, 173 grams of sugar inside of it. I don't know how successfully the yeast will be able to break that down, and even if they do, I don't know how successfully the carbon dioxide gas will be able to get out. Now, this is cheap, really runny syrup, so I'm hoping that it gets a pretty wild, or at least pretty unique reaction. The final one is going to have apple juice in it. Now, I believe apple juice, the sugar in it is sucrose, but it may be a combination of sucrose and glucose. I'm not 100% sure. The label doesn't tell me. I Googled it and I didn't find any definitive answer either. There will be 200 milliliters of apple juice in that flask, and I checked the label and there will be about 23 grams of carbohydrate in that beaker, so very similar to these 3 flasks. With the magic of video editing, I will really quickly set all of this up. I'm going to dump the yeast in this flask first because the yeast will not be able to sink into the syrup otherwise. Before we move on, what we want to do is try to, as best we can, dissolve the carbohydrates these are not carbohydrates, but dissolve them into the water as best we can so that the yeast have full access to those carbohydrates. So now I got to mix these up. I think that's about as good as I can get it. If I had more hands, this would be a little easier, a little bit more accurate. But the next step is to cover each flask with the balloon, and then we'll tip each balloon up so that the yeast gets added to these approximately all at the same time. The syrup one will be a little bit different, but that's a very different kind of setup. So I'm going to do that one last. What I'm doing right now is I'm squeezing each balloon to try to have as little starting air as possible in there. We don't want our lab results to be affected by user error, and I would be the user. Okay, these are ready to go. So we will add the yeast, and then we will let this run for approximately an hour, and we're going to compare how much gas has been made in each of these. So we need to swirl these again to make sure the yeast, it wants to float. We need to get it to the bottom. And as this experiment runs, periodically, I'm going to come over here and jiggle them a little bit to try to encourage as much fermentation as possible. So that'll all be in time lapse. Enjoy. Okay, we're gonna call the experiment finished at this point, and the results do surprise me a little bit. Now, let's try to hypothesize what's going on here. First, let's look at, well, let's rank these in order of gas production. So the flask with 10 grams of table sugar produced the most gas, and then second place, I'm gonna call these a tie. It looks a little bit like the apple juice balloon is a little bit bigger. So that had 23 grams of carbohydrate, I think fructose or sucrose, not 100% sure. The 10 grams of flour is in third place. Fourth place goes to 20 grams of flour. So more carbohydrate equaled less gas. I have an idea for why that is. The 20 grams of table sugar balloon, that also, so that goes in fifth place. Now I'm pretty surprised that this produced less gas than the flour balloon, since this one clearly produced more there. It could be that I had a tiny leak here. It could be that there was a tiny bit of air in there. Now, in a good experiment, or in a better experiment, I should say, I would have multiple of each of these items, but I don't have that kind of money right now, unfortunately. Now, our control balloon has no gas in it whatsoever. That is to be expected. There was no source of food in here, and no gas is produced. No real metabolism of any kind went on in that flask. Now, the artificial sweetener. These should be identical to our control group. This one has a little bit of gas in it, which is surprising. What could have happened is this yeast has a coating around it, and I think that coating is technically digestible by the yeast, so perhaps a little bit of gas was produced from that. But let's talk about artificial sweetener. It has no calories. It's not food. Now, the chemical tastes sweet to us, but that's just sort of a trick that it plays on us. It mimics sugar, it mimics carbohydrate, but not only is it not a carbohydrate, it's not food. It's like salt. You can't get any calories from salt or vitamins. They have no caloric value whatsoever. Our bodies can use those. Artificial sweetener goes in your body, and we don't metabolize it, break it down at all. So there are 0 calories, no work is done, no energy can be made from this. Now, let's talk about why less carbohydrate produced more gas than more carbohydrate. Here's what I think happened. I think what's going on is in these flasks, they're still busy breaking down the sugar and the flour. Because there was more food available, the yeast did not jump into fermentation until after these balloons or these flasks did. These flasks quickly consumed all of the available food, converted the glucose and the starch, which I believe is what's in flour, into pyruvic acid. And then once they ran out of glucose or starch to break down, then they started converting it into, what's this called? Acetaldehyde and ethanol and carbon dioxide. These flasks were behind because they still had glucose available to break down. That's my theory. That's my hypothesis, I should say. Oh, I forgot about the syrup. I had never done this before. I really thought that this yeast would be able to break down the syrup, but for whatever reason, it just simply can't. There's really nothing going on in there. It could be that my yeast are dead. Who knows? The syrup may have killed them. So, one other thing that I would like to talk about, why is this balloon larger than this balloon? Now, I think eventually both of these balloons would inflate to approximately the same size. And I think over a very long period of time, the balloons with more carbohydrate in them would get bigger than these two. They would just have to sit here for a long time. I believe that the balloon on the sugar is larger than the balloon on the yeast, or I'm sorry, the balloon with the flour, because put sugar in your mouth, and what happens? It dissolves very quickly. If you were to put flour in your mouth, it doesn't dissolve nearly as quickly. Glucose, table sugar, is broken down much more rapidly, so the yeast inside this flask converted the available carbohydrate into pyruvic acid much sooner than what was able to happen in this flask. So that is why this one, basically, it has a head start. I think eventually, because they both had the same amount of mass of carbohydrate, these balloons will get to the same size at some point. And these balloons, I want to believe, would get bigger than these ones over time, but they're still busy breaking down available carbohydrates. Hey, I let these sit for an additional hour and a half, so let's talk about what we have here. The control flask, still absolutely nothing happened here. We got some spillover into the balloon, but the 10-gram flask, 10 grams of sugar, has gotten quite large. It's pretty much the same size as the apple juice flask and the 10-gram flour flask. Now, the 20-gram sugar flask, it did grow, but it never got as big as any of the other ones. Whereas the 20-gram flour, not as large as the 10-gram flour, but significantly bigger than the sugar. Now, that's pretty unexpected. The syrup flask, yeah, still very disappointed in that one. Nothing ever came from that. Now these two, I'm also very confused about exactly what's going on here, because this artificial sweetener should produce no gas whatsoever. I've done this experiment before where it doesn't, but we do have a noticeable amount of gas in there. The control group, nothing. These should have matched that. So I don't have an explanation as to what caused that, but that's science for you. So I believe that's everything I wanted to cover. That concludes this lab. Thank you for watching.